So we define a number which shows the chromosomal location, okay, for each nucleotide. For example, if we want to say this nucleotide and address exactly this one, so we say chromosome 1, okay, and the location might be, for example, 50,563,123, okay? So this is the way that every nucleotide is being addressed in human genome. So altogether, as I said, we have these 2 billion nucleotides, more than that. And then in the human genome, the reference genome that we all basically use to be able to talk together about the human genome, every specific nucleotide has a very defined position and a number which shows that nucleotide. So that's why, that's the way that we can always address and talk about the specific nucleotide. We may say that in that specific position, what is the genotype of, for example, him? What is my genotype? What is another? How we express this? So we say it this way. In this position with this number, for example, this person has an A, and another person may have a C here. Okay, so this is the way that basically we express the position of nucleotide on the genome. All right, but when you look to these basically 2 DNA here, if we, for example, check here, you can see that we have CG here as well as CG here. So you know, in every double-stranded of the DNA, we have sense and anti-sense strand. One of the strands is our reference strand to say the nucleotide. So definitely on the other strand is the complementary nucleotide, which is present, okay, opposite that. So for example, we say that in this position we have G. So what we mean is the G is in the sense strand. On the other strand, obviously, is C. Someone will say that, okay, in this position we have C on the other strand. Okay, again, it's correct, because these are complementary. So, but whenever we want to say, we always use one of these strands to say that which nucleotide is present. So indirectly, obviously, we are basically presenting the other nucleotide as well. For example, we say, okay, that at this position there is an A. So it means, okay, obviously the complementary T would be present also. Okay, so for example, let's say this is the reference strand that we want to say the genotype for this specific nucleotide. So what would be, and if these are the 2 chromosome 1, for example, my 2 chromosome 1, then what would be my genotype at this position? Would be A. So it means that in this chromosome I have A and complementary, which should be T, and also here I have A, and the complementary strand would be T. But as I mentioned, we only say one of the strands. We don't need basically to say the others. So the genotype at this specific position would be A. It means that this person is a homozygote for A nucleotide at this position. Am I right? But sometimes you see that you say that, okay, someone might be in a state of being AA, might be AC. So what does it mean then? It means that in one chromosome it has A on the reference strand and T on the complementary, and the other chromosome it has C again on the reference strand and the G obviously on the complementary strand. But this is exactly the same position. So then it's possible then to have 2 different nucleotides, okay, exactly at the same position on different chromosomes. Is that right? And that is basically the genetic changes or the genetic variants which produce different alleles. Okay. So although this location, if this location is a gene, the gene is exactly the same, but these A to C changes produce 2 alleles, 2 different forms of the gene. And this change sometimes may have some functional consequences. These functional consequences sometimes are just normal consequences. It creates some normal features. It creates some differences, but these differences and polymorphism just create just normal differences which is present between humans. But sometimes these changes might be deleterious. which may affect the function of gene. For example, it may completely abolish the gene function. Am I right? Yeah, this is possible. For example, if this A changed to C, it may basically put a new amino acid in the position of the normal amino acid, and this amino acid obviously may interfere completely with the normal function of the protein, which is being basically translated from this gene. Then, so the result of my basically explanation is this. Sometimes these changes in the nucleotide are normal polymorphism. So if you see changes from one nucleotide to another nucleotide, there is no problem. Okay, it only creates basically normal differences between human features. But sometimes these changes are deleterious. They cause problems. They may cause death, even embryonic death. Or sometimes they may end up with a child or with a human with a genetic disease. Okay? So now the question is this, that what is the importance of these different nucleotides in terms of these changes? Is it the same? For example, if this change happens in any of these nucleotides, do we get the same result? No. Some nucleotides, if they change, is fine. We don't have any problem with these changes. Basically, our physiology tolerates these changes. But for some nucleotides, these changes are not tolerable. If it will happen, it definitely creates a disease. Right? And how do we express this? So the way that we express, okay, these our tolerance towards these changes is to say that how conserved basically is a nucleotide, or a nucleotide maybe. Some of the nucleotides or some of these positions are very, very conserved in human, even into the basically if you look to the other species, you again see that they are conserved. You know, for some genes or for some sequences, if we start from prokaryote to come to eukaryote to the human, you will see exactly the same sequence. So they are very conserved. You see exactly the same nucleotide at those positions. So if you end up, or if you see such a thing, if you see some very conserved nucleotide among all different species, so what is the result? So what you can elucidate from this? You think that these nucleotides or these sequences probably are very, very important. During evolution, you never see changes in these sequences. So what happens if you accidentally, or if you check a patient or an embryo and see changes in these nucleotides? So this should be a red flag, basically an alarming function. Okay. So if you have a sequence which you already know that this sequence is very conserved among all different species, among all humans, whenever you see a change in these conserved sequences, this will put a big question mark for you. So this might be a deleterious change. Okay? And on the other side, sometimes you see that, okay, some sequences are really changeable. So you see that different species has different nucleotide at that position. So you feel that, okay, it looks like changes of nucleotide at this specific position probably is tolerable. And that is responsible for some minor, basically, changes or differences that we have in normal human features. And probably is not deleterious. So conservation of the nucleotide and the gene sequence is a very important issue. Okay, when we want to basically interpret a genetic change, to say if it's basically pathogenic, if it causes diseases, or no, it doesn't cause diseases. Okay, so that is very important. Okay, so now let's go back to this structure that we have here, and then I focus to this specific nucleotide. You know that this specific nucleotide, which is A in this chromosome, and also A in this chromosome, has a specific position. So now the question is, do you think at this specific position, we always have A. So if we have, for example, this 60 chromosome 1, 120 chromosome 1 that we have in this classroom, and if we check this position, do you expect that we always get A here? Okay. We may get A in all these 120 chromosomes. We may get A even in all 180 million chromosomes that we have in the body. Am I right? For some nucleotides, this is the case because they are conserved, as I mentioned. And we call them monomorph. We say that this nucleotide at this specific position is monomorph. There is only one form of that in all human, basically, chromosomes. But sometimes this is not the case. If you look, for example, to these 120 chromosome 1 here, 100 of these chromosomes may have A and 20 may have B. May have C for it, not B. Okay, so 20 of them may have C. So what we say here, we say that this position is a polymorph. It's not monomorph. Because we already saw that in a population of 120 chromosomes, there are 100 A and 20 C. And if we look to this as a specific population, then we can calculate the frequency of these alleles. So these A and C call two alleles for this position. So then we say that this specific position which has a big number, which basically characterizes that, has two alleles. One allele is A and the other allele is C. And if we look to a population of the human, for example, this population here, 60 individual, 120 chromosome 1, then we expect to have 120 alleles. And from these 120 alleles, 100 of them are A and 20 are C. So this is the concept of allele and then allele frequency. This way, we calculate the allele frequency in different human populations. Okay? So probably you hear already, or you basically, when you read the book or an article or something, then you, many times or frequently, you see that they talk about allele frequency. So what is the concept of allele frequency? That is the concept. So every human population has a gene pool, or basically every population has a gene pool. So what is the concept of gene pool? So if you look all the genes from the members of that population and put it in a pool, that's called the gene of that population. So the gene pool may consist of all the genes, basically all chromosomes and all genes. Or sometimes you may describe this gene pool for a specific gene. For example, say the gene pool of the population in this classroom for beta-globin gene. So it means that you get all the beta-globin genes from these people and put it in this pool. This is called the gene pool of this population for beta-globin. And this might basically the same thing you may create for every single gene that we have. So what we have in this gene pool? So every one of us has 2 copies of beta-globin. And if we check, and if we get all these beta-globin and put it in one place, then we have all different form of beta-globin in this. So basically all variation which is present in beta-globin and all the alleles of beta-globin will be present in this gene pool. So then in this gene pool, we can basically check that different forms of beta-globin has which sort of frequency. And this is called allele frequency for these different alleles or different form of beta-globin. So the same thing may happen even for a single nucleotide. For example, for this nucleotide here, we can say for this nucleotide, this gene pool consists of 120 alleles. 100 of these alleles were A and 20 of them were C. Therefore, what is the allele frequency? So the allele frequency for C would be 20 over 120. All right? So basically 1 over 6. And the frequency for the A allele would be 100 over 120. Then obviously it's possible to express that as a percentage or a fraction that I mentioned, 1 over 6. So this gives you the allele frequency